So it's a great pleasure to welcome Dr. Danzer from University of Cincinnati today. He is my collaborator on a project that I kind of drifted towards studying the role of effectors of the mTOR pathway in epileptogenesis. And while looking for somebody who could teach us how to do EEGs and such, I discovered that we have a very accomplished neighbor not far away who actually made a big impact on the field, discovering role of neurogenesis and mTOR in its newly generated neurons driving development of epilepsy. And that's, I guess, what we'll hear about. So Dr. Danzer comes from did postdoctoral training in a very famous laboratory in epilepsy of Dr. McNamara at Duke. He did PhD at University of Arizona. Got a very robust program going at the University of Cincinnati. I think started there as a faculty in 2005 and got very impressive track record since then, with a major accent on neurogenesis. But he also studied some other weird things like anesthesia-induced cell death. Got recently a very exciting paper on that, demonstrating that age of newly generated cells actually counts in the sensitivity, determines the sensitivity to anesthetics. So I'm not sure we'll hear about that too much, something you can look it up and read. And the floor is yours.
All right, can everybody hear me yet? There we go. I'm going to move this up a little bit. Thank you very much for the kind invitation and introduction. So the first slide I put up here is just sort of a reminder for me. So I'm a basic scientist, but this is a representation of a woman with epilepsy, and the seizures here are represented by butterflies. And I really like this sort of imagery because in the epilepsy lab with our mice and rats, when they have a seizure, we get excited. But I think it's good to remember that for the patient, these are a bad thing. Okay, so I'm going to start by just running down sort of a quick summary of what I'm going to talk about today. So the big question that my lab focuses on is trying to understand how the brain becomes epileptic. And the hypothesis that we've been pursuing is that accumulation of abnormal neurons in the hippocampus is playing a role in the development of temporal lobe epilepsy. So the evidence that I'm going to talk about today that suggests that might be the case first comes from studies looking at the anatomy of these hippocampal granule cells. And I'm going to show you that they actually do integrate into the brain abnormally in epilepsy. We did some studies looking at the sort of the load, or how many of these cells are present in the brains of epileptic animals, to determine whether that correlated with their disease severity. I'm going to talk about some work we did again with the mTOR pathway, trying to get at the question of whether these abnormal cells are actually capable of causing the disease. And then I'm going to talk a little bit about some of our future work looking at epilepsy progression. Please stop me at any point if you have questions. I like to keep it interactive. So I think I have to go up here to run this. So first, I just wanted to orient everybody on the structure we're looking at. So sort of up in the upper right there, you're looking at a coronal section of a mouse brain, and the box in green is the hippocampus. And what you're going to see here is a 3D reconstruction of the dentate gyrus in the hippocampus. So this structure is kind of a V-shaped structure, so the cell bodies are located here. There's a cell sparse region in the middle where all the axons live and a lot of interneurons. And this structure, the dentate gyrus of the hippocampus, is thought to be very important for learning and memory. Damage to this structure causes memory deficits in humans, and we're going to talk a lot about one of the unique features of this cell population is it produces neurons throughout life. So it's one of the few regions of the brain where you have adult neurogenesis occurring. I don't need to go back. So And these adult-generated cells are the ones in particular that we think are responsible for contributing to epileptogenesis. So what you're looking at here is a circuit diagram of the hippocampus, and this is complicated and it's actually simplified, so it's even more complicated than it's represented here. But the main reason I wanted to show this is just to demonstrate that this structure is sort of organized as a loop circuit. So here you have neocortex, which feeds into entorhinal cortex. This feeds into dentate gyrus here, which then feeds information that sort of goes around the hippocampal circuit, and then it comes back to entorhinal cortex. And the reason I show this is that I think this sort of circuitry may be one of the reasons the hippocampus is sort of notorious for being seizure prone. Temporal lobe epilepsy is one of the more common forms of epilepsy, and it frequently involves this circuit. And again, if you sort of imagine that if you don't have good inhibitory control over this circuit, so you have information coming in, that's going to feed back into the outputs, which gives you more information coming in, and you could easily get sort of a runaway process where you get seizures occurring. So there's a lot of mechanisms in the circuit to try and prevent that from happening, and one of these is thought to be the dentate gyrus. So this is our structure of interest here, and what it's thought to do is sort of act like a gatekeeper. So a lot of information comes into the hippocampus from entorhinal cortex. Very little of that information actually makes it through to the next cell population, the CA3 pyramidal cells. So the dentate gyrus sort of stops a lot of that activity from getting through in the normal brain. In epileptic brains, there's been a number of studies now showing that this sort of gating function of the dentate starts to fail, and you get much easier transmission of activity through the dentates and through the circuits, and that's hypothesized to contribute to epileptogenesis. So one of the reasons that we think the dentate might fail in epilepsy is because of accumulation of abnormal granule cells. So I'm going to give you a little bit of neuroanatomy real quick here. So in red is sort of the normal structure of a granule cell. So it's got a cell body here, it's got dendrites that come out one side of the cell, and an axon that comes out the other side of the cell. And that's sort of the normal morphology of the cell. So in epileptic brains, in animals and humans, we have a number of pathologies that have been known about, some of these for a long time. So the first of these is termed mossy fiber sprouting. So this red fiber here is called a mossy fiber axon. And in blue here is shown what happens in epilepsy is these axons project back into the dendritic field of the granule cells and actually form excitatory synapses with these granule cells. And that creates what's been termed a recurrent excitatory loop. So this cell population now excites itself. And that's, for obvious reasons, thought to be a problem if you have a disease like epilepsy where hyperexcitability is sort of the key feature. So more recently, we found basal dendrites on these cells. So normally they don't have basal dendrites, but in green here you have this basal dendrite, and these project into the axon field of the granule cells where they also get innervated by granule cell axons, and you get more of these recurrent excitatory loops. And then you have some cells that migrate, so they're supposed to live here in the cell layer, and the cell in yellow here is a granule cell that's migrated the wrong direction out into this, what should be a cell sparse region. And because it's in the wrong spot now, these cells have all the wrong kinds of contacts, and physiological studies on these cells suggest that they're hyperexcitable and have a number of unusual properties. So if we jump back to our circuit here, you can sort of see it under the black box here. In the epileptic brain, you have sort of formation of a new pathway here shown in red, where this dentate granule cell population is now exciting itself. So you have the formation of these recurrent excitatory loops. And that formation is hypothesized to contribute to sort of this failure of the dentate gate, destabilizing of the circuit, and that's thought to promote epilepsy. Okay. So I mentioned that one of the unusual things about granule cells is that they're generated throughout life. So this is one of two populations that's produced in adulthood. The other is olfactory bulb neurons. These newly produced neurons are thought to, again, be important for learning and memory. So if you disrupt neurogenesis in animal models, they have poor performance on maze tests and things like that. So what you're looking at here is a fate mapping strategy that we use just to look at these newly generated cells in an adult animal. And what's shown here is a progenitor cell. So this is a type 1 progenitor. it produces type 2 progenitors shown here. And these produce immature granule cells, which then go on to produce these mature granule cells. And this particular animal had epilepsy, so you have an ectopic cell here, and then a cell with one of these basal dendrites here. So one of the first things we did when I started my lab was to try and establish where these abnormal granule cells were coming from. So we knew they were there. We knew adult neurogenesis occurred in the dentate, and what we hypothesized that maybe the abnormal cells are these newly generated granule cells. And that sort of made sense. We know that a newly generated cell is going to be more plastic and more capable of making changes than a mature cell. We also know that from studies of developing animals that developing neurons are much more sensitive to brain insults than mature neurons, which is why that a child exposed to excessive alcohol, you get fetal alcohol syndrome. In an adult, same exposure, you might be sick the next day, but you don't have permanent brain damage. So these young neurons are particularly vulnerable to insults, and our idea was that maybe this epileptogenic insult is disrupting the development of these cells. So what you're looking at here, sort of cut off a little bit, is a birth-dated granule cell. So this cell was born in an adult animal about 1 week before we treated it with a drug called pilocarpine that causes it to have status epilepticus, and then this is an animal model of epilepsy, so these animals will go on to develop spontaneous seizures about a month later. So as I said, this cell was born a week before the insult, so it's an immature cell at the time of this brain injury, and we looked at it a couple months later. So the blue label is this BRDU birth dating label that we know this cell was born at the time that we injected this drug to birth date them. And what you see, this particular cell, so this is the axon of this cell coming out here, and coming down this way is where these axons should be going, and you see this cell has an axon going the opposite direction, where it's potentially going to make excitatory contacts with other granule cells shown here and create these recurrent excitatory loops. So we also used a similar strategy to look for these ectopic cells. So you can see these blue labeled cells here. So these are the newborn cells in the epileptic brain. So they're supposed to live here in this cell body layer, and you see that they're out here in this hilar region where they don't belong. So these are these ectopic abnormal cells I talked about, and here's another cell, BRDU birth-dated cell with these basal dendrites. So through our work and work of a number of other labs, we've been able to establish that a lot of these pathologies are produced by these newly generated cells. So I'm going to show you another example. So this is a different epilepsy model, and one of the things we wanted to find out was this specific to just one type of epilepsy in these animal models, or does it affect a lot of different models? So this is the intrahippocampal kainic acid model of epilepsy, and these are granule cells. So the color scheme is a depth filter applied to them. So parts of the image that are in different depths of the tissue are different colors to make it to reveal the cells a little better. And in this particular model of epilepsy, we also found basal dendrites. And I'm going to start this little movie here. But these cells were really unusual. You've got these two cells here that have these long basal dendrites, and these particular cells have no apical dendrites at all, which was, very unusual morphology. So now, if you think about the cell, which is supposed to receive all its input from the apical side, it now has no apical dendrites, so it doesn't even have its dendrites in the right spot to receive that input, and now it's getting all its input from the basal side of the cell, which are likely mediated a lot by just neighboring granule cells. So these cells we think are probably particularly pathological and maybe contributing significantly to these recurrent circuits. So another pathology that we've seen, which is going to become important as we get into the mTOR part of the talk, is hypertrophy of some of these newborn cells. So you can see that in these images here. So these are all newborn granule cells. This is birth dated using a fate mapping technique, which I'll tell you about in a minute. And you see this cell here is significantly larger than these neighboring granule cells. And here's another example. So you have a newborn granule cell that's much larger than its neighboring cells, and that one's cut off a little bit, but this guy is a lot bigger than these two. And associated with this neuronal hypertrophy, these cells tended to have basal dendrites and also very thick apical dendrites. So this is, in this case, we don't think this is sort of an unhealthy cell in the sense that it's dying or something. We think this is sort of a cell that's kind of overgrown, so it has a lot more spines on it. It's probably receiving more input. And one of the ideas in the epilepsy field is there may be what's called hub cells, which receive a lot of input from neighboring cells, and then they're talking to a lot of cells. And these hubs potentially may be able to sort of drive epileptogenic activity. Okay, so to kind of summarize what we learned from these birth dating studies, I've sort of condensed this into this schematic here. So the way to read this, so this red bar is the onset of status epilepticus in the animal. So this is this brain insult that makes the animals develop epilepsy. And what we did here is we looked at different populations of cells born at different times prior to this insult. So we've got mature cells, which are greater than 7 or 8 weeks old. So these cells have already differentiated and sort of they've completed migration and their axons are grown. And what we found is when you expose these cells to an insult, they don't change very much. So we've seen some reductions in spine density, but they don't migrate to the wrong spot or grow basal dendrites or things like that. As you get younger and younger cells, so you get more and more severe pathology resulting. So what we call juvenile cells are about 4 weeks old, develop these hilar basal dendrites. Even younger cells can develop these hilar basal dendrites and also have apical dendritic abnormalities. And then the most severely affected cells are those born after the insult. So sort of the entire life of the cell is in this now injured brain. And these cells are the ones that migrate to the wrong locations, and they contribute to the other pathologies as well.
If you were to block, use some kind of block step gene or something driven off an ST motor, and you could block immature or newborn cells right before you induce. Can you stop epilepsy from occurring?
So we're trying to do that experiment. I'll talk a little bit about that, some of the strategies that we have. What some other labs have done that have produced some really interesting data is to use anti-neurogenic treatments like Aracee to block neurogenesis. And they found, so there's a couple papers that found that when you do that and reduce neurogenesis, you get a reduction in seizure incidence in the animals later on. So that's really promising. So there's some other papers that didn't find the effect, but there's a lot of caveats with those as well. So with these drugs, they can cause a lot of other problems. So,
Steve, has anybody treated these types of cells with agents that cause excitatory neural activity but won't necessarily cause seizures per se? I mean, clinical seizures in the animals?
To see what effect that has on them?
Yeah, to see what, so in other words, a subconvulsive dose of...
Right. So, I don't know if they've specifically done that experiment, so you can certainly affect their behavior with physiological conditions like learning, can promote the survival of these new cells. So you can activate these new cells so they'll express CFOS. With milder insults like the Kinley model where you induce seizures, but the animals don't develop epilepsy, you also get some changes in this cell population that suggest they may be more hyperexcitable. So that somewhat gets at your question. I don't know I'm not aware of a paper where they've sort of applied a systemic drug that would just shift it a little bit. But
yeah, I'm just thinking about your PTEN model because, in PTEN in humans with mutations, they don't always necessarily develop epilepsy or / clinical seizures, but they have obviously all this abnormal behavior, autism behavior. So
right.
the question is the degree of which neural activity occurs within these populations. How does it affect structure?
Right. So, I think that's what you're getting at is a good point, is that epilepsy may be one manifestation of having these abnormal cells in the brain, and it may be that they also contribute to some of the comorbidities that are associated with epilepsy. So epilepsy has a really high incidence of depression and can have cognitive problems as well. So disrupting these cells could be important in that way. And you could certainly imagine that you have abnormal cells in patients or animals that isn't quite sort of the threshold they're having overt seizures, but it's still disrupting function. So so they might have learning deficits. So one other point on your question is that the experiments to block neurogenesis that have been tried so far blocked neurogenesis here, right? And nobody's tried blocking it, so I think you'd really want to block it here so that you take out these populations of abnormal cells too. So I'll talk a little bit about a strategy we have to try and get at that. Okay, so this is just a quick schematic of one of the fate mapping systems we use. So this is a Cree-Lox system that allows us to permanently label these newborn granule cells with GFP. So we use 2 mice for this one. So we have a mouse that drives Cree recombinase driven by the GLI1 promoter. And this particular form of Cree recombinase is only activated when you inject the animal tamoxifen. And if you're unfamiliar with what Cree recombinase does, basically you can think of it as sort of a DNA scissors. So when it's activated, it'll look through the genome and it finds these sites called LOXP sites, and it just splices out whatever is in between. So we cross this animal to a second animal that has beta-actin-driven GFP, but it's got another gene, colon acetyltransferase, sort of in between here, and a stop codon. So without recombination, you don't get any GFP expression, but when you activate the Cree recombinase with tamoxifen, it cuts this out and the cells turn green. So the GLI1 promoter here targets it to neuroprogenitor cells. So what we do for a lot of these experiments is we wait till the animals are a couple weeks old, give them tamoxifen, and the combination gives us pretty good specificity to where the only cells we're targeting are these adult-generated neurons. So using this approach, we wanted to ask first a fairly simple question. So if our hypothesis is correct and abnormal granule cells are contributing to epilepsy, there should be a correlation between the number of abnormal granule cells that are present in an epileptic animal and the severity of its disease. So we predict that the more abnormal cells the animal has, the worse its disease is going to be if our hypothesis is true. So we did that using this fate mapping approach, and we took advantage of the variability in disease severity in this animal model. So we have some animals that have very mild epilepsy relatively. So this animal had 0.1 seizures per day, or 1 seizure every 10 days. This animal had very severe epilepsy, so 3 seizures a day. We looked at the brains of those animals, specifically looking at these newborn cells, and what we found is that animals with few seizures had very normal-looking brains, whereas animals with lots and lots of seizures had very abnormal-looking newborn cells. And when we correlated that, looking at these ectopic cells, we found significant correlations between the number of seizures per day and the percentage of new cells that were ectopic. We also looked at this mossy fiber sprouting and found a similar correlation, and we found some trends with the basal dendrites. So that doesn't prove that abnormal granule cells cause epilepsy, but it's consistent with our hypothesis, and if we'd found a negative trend or something, then we'd start to think maybe we're on the wrong track. But the fact that there does appear to be a correlation between the number of abnormal cells and disease severity, this makes us think that maybe we're on the right track. So to kind of summarize where we are so far, so we know that the epileptic brain has lots of abnormal granule cells. We now know that most of these abnormal cells are derived from these newly generated or adult-generated cell populations, and they have a lot of abnormalities that are at least consistent with the idea that they may promote hyperexcitability. So these basal dendrites and the mossy fiber sprouting and so forth, all by creating these recurrent circuits, might contribute to epileptogenesis. And we've also shown that the load, sort of the number of abnormal cells you have, predicts seizure frequency. So kind of the big questions now, and what we wanted to focus on next, is do these newborn cells really cause epilepsy? And kind of the 2 classic questions that you ask if you want to know if something causes the disease are A, is the particular phenomenon necessary for the development of the disease, and is it sufficient for the development of the disease? So kind of restating the question, it's sort of a chicken or the egg. So we know abnormal granule cells are in epileptic brains. It could be that they cause the epilepsy. It could be that the epilepsy in the animal is causing these abnormal cells, and it's more of an epiphenomenon. So to get at that, I'm going to sort of switch a little bit and talk about the mTOR pathway. And the first question we tried to address was the sufficiency question. So are abnormal granule cells capable of causing epilepsy? And we focused on the mTOR pathway. It's an interesting pathway. So it's one of the things it does is it regulates granule cell development, but it's also a pathway that's looking to be very important for a number of diseases, including epilepsy. So what I've circled here, so here's mTOR. So this is mammalian target of rapamycin. It's a pathway that regulates neuronal growth and cell survival and a lot of important cell functions. And these genes or parts of the pathway that I've highlighted are genes that are known to cause human disease. So this gene here is TSC1 and 2, so tuberous sclerosis complex. So when kids get this disease, they get a lot of tubers in the brain. A lot of these kids develop autism, and a significant percentage also develop epilepsy. So BDNF and NMDA receptors are also linked to epilepsy, and relin and NF1 are a couple other genes that sort of feed in this pathway that are linked to human diseases. The schizophrenia gene, DISC1, feeds into this pathway, so there's potentially a link there. And there's a number of other ones that I'm not showing. But this particular pathway is looking to be a very interesting pathway for a number of neurocognitive diseases in humans. So to sort of explain the epilepsy focus of this pathway, so one of the things we know about the mTOR pathway is that during the development of epilepsy, this pathway is activated, and it's activated very robustly. So one of the markers for mTOR pathway activation is staining for, go back here, S6. So where is it? So this is S6 kinase, so it gets phosphorylated when mTOR is activated. And so this blue stain here is staining for phospho-S6. And this is an animal that got kainic acid injected in one hemisphere of the brain, and that causes seizures in the animal. And these animals will go on to develop epilepsy. And what we're looking at here is the two different hemispheres. So this is the uninjected hemisphere, and you see very little activation of phospho-S6. This is the same animal, but the injected hemisphere, and you see all this blue staining here. So it really robustly activated this mTOR pathway, and then associated with that, if you look at the actual granule cells, so this is the uninjected hemisphere, and these granule cells look pretty normal. This is the injected hemisphere, and you can see that these granule cells are much larger. So you remember we talked a little bit about hypertrophy of the newborn cells. This causes really massive hypertrophy of many granule cells within the hippocampus, and you also get spreading of the granule cell layer, so some dispersion. And this somatic hypertrophy, so cell swelling is one of sort of the characteristic features of excess mTOR activation. So the cells grow too much and they get too big. So we think that this phenomena here is probably related to the increased mTOR activation. So there's a number of labs that have also looked at the effect of blocking mTOR signaling during epileptogenesis using this drug called rapamycin, which is this mTOR antagonist. So this is Michael Wong's work at WashU in St. Louis. And what they did is they treated animals with rapamycin and looked at seizure frequency. The black bars are the untreated animals, and you see that they're having quite a few seizures. You can barely see the treated animals here, so this had a pretty dramatic effect in this model of controlling seizures. This is work from Paul Buckmaster's lab, where he's able to show that if you infuse rapamycin, you can block this mossy fiber sprouting. So you can actually see this black band of staining here. So this is this mossy fiber sprouting that's sort of very characteristic of temporal lobe epilepsy. And when you infuse this drug, you can prevent that. When you take the drug away, it comes back, unfortunately. But that suggests that this mTOR pathway is maybe involved in this particular pathology. So our question we're going to ask now is that if abnormal granule cells cause epilepsy, if we take a normal brain and we add abnormal cells to that brain, the animal should get epilepsy. So the way we're going to do that is using this mTOR pathway, and we're going to selectively knock out a gene called PTEN here, which sort of acts as a stop on this pathway. So this gene is actually also mutated, and there's kids with PTEN mutations who have neurological disease. So the reason we pick this gene, so you knock this gene out, you get excess activation of the mTOR pathway, and that does a couple things. So it makes the granule cells very abnormal, and it makes them abnormal in a way that's very similar to what you see in epilepsy. This is just sort of back to the transgenic model we used to do it. So you've seen a lot of this before. So we're again using this GLI1 promoter, which drives expression of Cre recombinase in neural progenitor cells. So here's our progenitor cells in green here in the hippocampus. So we're going to turn these cells green as before, but in addition to that, we're going to cut out exon 5 of the PTEN gene. So what that will give us is a small percentage of granule cells that have no PTEN. All right? So this is what that actually looks like. So the green panel at the top here is new N staining. So this just labels all neurons. Then PTEN staining in red. So in these panels, this is a control animal. And you see that basically all the neurons, I think I just lost my...
Sorry, lights still on.
Turn it off? Is that right?
Yeah, maybe the battery. Let's see it. I'll see if I can.
Okay. I'll talk loud. So the normal cells here they all have PTEN. So basically all the neurons have PTEN. And if you look at our knockout animal here, see most of the neurons still have PTEN, but you see these gaps here in the staining, and these correspond to regions where you have new N staining. So there's a neuron there, but it has no PTEN. And that's what we wanted to produce, is this mosaic effect. Because we know that in an epileptic animal, not all the granule cells are abnormal. It's just a small percentage relatively. So we wanted to see what happens if we just make the small percentage of granule cells abnormal. So this is what the cells actually look like when you knock out PTEN. So here's two normal granule cells in one of these animals, the cell body, very thin dendrites. And this is a PTEN knockout cell. So these things sort of become monsters. So you get this really dramatic hypertrophy of the cell body, very thick dendrites, a lot more spines on those dendrites. So these spines are points of synaptic contact with other cells. So more spines suggest they're receiving more input. So very abnormal looking cells, but if you remember some of the images I showed you earlier, particularly this hypertrophy, that's similar to what we see in some of our traditional forms of epilepsy. So I'm going to play a movie for you real quick. So this is a image from a brainbow mouse. So these are all PTEN knockout cells, and you can see as this sort of rotates around.
It's off. The batteries work, so you can put it on, turn it on.
Okay. Go. All right, so we have ectopic cells here that this PTEN knockout was able to replicate. And you can also see a little patch of mossy fiber sprouting. So this patch here is actually an axon that's coming in here and going back into the granule cell layer. Back online here. Okay. So using this approach, we're very happy that we were able to produce abnormal cells that looked pretty similar to what we see in some of these epilepsy models. And so this is just another image. So one of these ectopic cells here, basal dendrites on these cells as well. Again, similar to what we see in epilepsy. Okay, so some of the other pathological changes we've seen with this PTEN deletion is looking at the sort of the axon pathway of these dentate granule cells. So just to orient you, here's the dentate gyrus. So here's this cell body layer. This is the region where the ectopic cells live. And shown in blue here is where the axons of these cells project. And these granule cells, so they're unusual for a lot of reasons, but they have a very unusual synapse called a giant mossy fiber bouton. And this is one of the largest and most potent synapses in the brain. So these things can actually be about 5 microns in diameter. So that's huge compared to, say, cortical synapses, which are going to be less than a micron. And they contact these unusual postsynaptic structures called thorny excrescences, shown here on a CA3 pyramidal cell. And this synapse is referred to as the detonator synapse because you can activate a single granule cell and it can fire its target pyramidal cell. So that's really unusual. So say for a cortical neuron, it might need simultaneous input from hundreds or even thousands of neurons to get it above threshold to fire. And in contrast, you've got a single granule cell which can fire its target pyramidal cell. And again, that may be one of the reasons that this circuit is so seizure prone, is because you have this just really powerful synapse. So when we looked at the axons of these cells, we found two changes. So first, so this is the PTEN knockouts here. The axons were much thicker. So that would suggest they might have better conduction velocity. Curiously, we also found that they had fewer of these giant boutons. So that's somewhat paradoxical for sort of increased excitability. So it suggests they're maybe contacting fewer pyramidal cells. We haven't looked at the full extent of the axon yet, so they may be longer and they may compensate for it by increased length. But so far, it looks like they may contact fewer cells. But again, the thicker axons may make these axons more efficient, so they're able to have less sort of failure of the signal going down the axon, just like a thicker cable if you think of power lines or something like that. So that's quantified over here, but greater axon diameter, but again, fewer boutons for these cells. Okay, so kind of the big question here is, so we got these abnormal cells in the animals. They look abnormal in ways that are similar to epilepsy. The question is, do the animals actually have epilepsy? So what I'm going to show you here is an EEG recording from one of these PTEN knockout animals. It's going. And the way we record these guys is, so they have EEG electrodes in cortex or hippocampus, and then there's actually a radio transmitter implanted under the back of the animal here, and then it sends the signal to this receiver plate, so it's an entirely wireless system. And if there's any neurologists here, you probably recognize that's the onset of a seizure. And it's a
time frame?
So this is, I think it's about real time. But the whole seizure is going to last about 30 seconds to a minute or so. I'm sorry, we don't have the scale up here. Still playing with movie making. But you see the animal here. So this starts out, you can't tell he's having a seizure much, but then it goes into sort of a typical mouse convulsive seizure as it spreads to motor cortex. And that continues for a little while, and it stops, and the animal goes into a theta rhythm, which is pretty typical. So what we found in these animals is not only do they have epilepsy, they have a really sort of robust, disastrous epilepsy. They have a lot of seizures, and these seizures appear to get progressively worse over time. And so a lot of these animals die prematurely, so 4 months or so old, we get a lot of drop-off of these animals. So one of the things we've been working on now, so in our initial studies, we knocked out PTEN from about 10 to 25% of the granule cells, and that produced epilepsy. So we wanted to sort of modify this model a little bit and see if we could also produce epilepsy by knocking PTEN out from a smaller number of granule cells. We really wanted to get it down to around 5% or so because we think that's similar to the percentage of abnormal cells that are present in an animal with epilepsy or possibly in humans with epilepsy as well. So we're able to do that by just altering the tamoxifen dose. So you give them less tamoxifen, you get less recombination. And we now have it to where these animals, so if you knock PTEN out of about 3 to 5% of the granule cells, they still get epilepsy. And this is an EEG seizure from one of these animals. This is recording from hippocampus. They have unusually long seizures in hippocampus. So again, we left off the time scale. Sorry about that. But this seizure is about 6 minutes long, which is so a typical temporal lobe epilepsy seizure might last 30 seconds, a minute or so, and then it stops. So in the hippocampus of these animals, we're seeing these very long seizures, which is unusual, and I think manifests to the kind of severity of the epilepsy these animals have. But what we did find when we reduced the number of abnormal cells in the animals is that mitigated the phenotype. So in our first study, we're sort of on average knocking PTEN out of about 15% of the granule cells. They start having seizures around 60 days or so. And when we knock it down to about only 3 to 5%, that delays the onset of the seizures by about a month or so. And then we had a couple animals that have very low recombination, so less than 1% PTEN knockout cells. And as far as we can tell, those animals don't get epilepsy at all. So it looks like there's a threshold effect that we think at some point here that you're sort of below a threshold, and having a few abnormal cells doesn't hurt you too much, at least with regard to epileptogenesis. So one of the things we want to do with this model now is look at epilepsy progression. So in some patients with epilepsy, their epilepsy can get more severe over time. And then as people are getting epilepsy, we think that it probably certainly starts before you have clinical seizures. So if you think of patients with traumatic brain injury where they have the injury, and then years can potentially go by before they have the first clinical seizure. So the question is, what's sort of happening in that period that we call the latent period between the injury and the first seizure? What sort of changes are undergoing in the brain of either the patient or the animal that causes them to eventually develop epilepsy? So we want to use this model to get at that question, and the unique feature of this model is that we know exactly where the epilepsy has to begin. It has to start with these PTEN knockout cells, because it's the only thing that's really wrong with the animal. And we want to start using that now to look and see how do changes spread from those knockout cells, what happens if you manipulate those cells at different time points in the disease. And again, we think this is very important because understanding this progression gives you some insight in what you might have to do to treat a patient with epilepsy. So how important it is. So if you say want to block neurogenesis to prevent epilepsy, how important is that you block that right away? Can you wait a couple of years after the patient starts having seizures and block it? Will it still work then? We don't know. We'd like to answer that question. And I'll show you in a minute some of the strategies we're going to use to try and get at that. So first, we do appear to have progression in this model. This is looking at, again, this 3 to 5% knockout and the onset of hippocampal seizures versus cortical seizures. And we would expect the seizures to start at hippocampus because that's where the abnormal cells are. And when we start recording from these animals, we have to wait until they're big enough so we can plant the transmitters, but they're about 6 weeks old or so at that time point, and we find that most of the animals are already having hippocampal seizures when we get the electrodes in. But when we record from cortex, it looks like it can take quite a bit longer, so even a couple months longer before those hippocampal seizures start recruiting cortex, and you have sort of spread of the epileptic focus. And then this is looking at an onset of hippocampal seizures in days, 60 days here, versus the first cortical seizure at about 100 days. So we want to know sort of what happens between here and here. What are the changes in the brain that allow that seizure to spread to cortex, which is, if you got seizures, you'd rather have more focal than spreading to your whole brain. So what we think we want to look at here is primary and secondary epileptogenic changes. So we think some of these changes are going to be mediated directly by these abnormal cells. Some of these changes are going to be these abnormal cells causing changes among other populations of cells. And we have sort of evidence already that the PTEN knockout cells are initially very abnormal. So we've done some patch clamp studies on these cells. And this is a normal cell. And this is a PTEN knockout cell. So you can see this morphological difference between the two. And just looking at these single cells, we can see that the physiology of the individual cells is very abnormal. So this is running a voltage ramp on these cells, which will cause them to fire. So these downward spikes are firing of the cell. And what we see in a normal cell is that it inactivates very quickly as you sort of ramp the voltage up, and the cell basically shuts down. When you look at one of these PTEN knockout cells, you see that it doesn't shut down. So you have almost no inactivation of the cell. And then also, sort of oddly, we see doublets with the first spike in a lot of these cells. And we think this may be an indication that some of these cells may be gaining sort of bursting property. So bursting cells have actually been seen in epilepsy, so we may be mimicking what happens in some of these other epilepsy models. So also looking at the trace here, so these downward spikes are just spontaneous excitatory postsynaptic currents. So this is a control cell. This is the knockout cell, so you see a lot more of these downward spikes, indicating that it looks like these cells are receiving a lot more spontaneous input. So we've also done some field potential recordings on these slices. So basically, you make a brain slice from the animal and you stimulate here and record here in the granule cell layer. So you're basically activating the incoming axons. When you do that, you get an excitatory postsynaptic current. So one of these shown in red here. And if you stimulate high enough, you start to get a downward spike, which is a population spike where the granule cells actually start to fire. So in normal tissue, you get, so as you increase the current, which is done here from blue to red to green, is increasing current, so the population spike or the EPSC gets bigger. Then you get above threshold, you usually get a single population spike. In our PTEN knockout slices, we get multiple population spikes, which suggests that as you activate the circuit, you may be getting sort of reverberatory signals going through it. And this is also characteristic of epileptic brains, where you get these multiple population spikes. So this model seems to recapitulate what you see in some of these other more well-characterized models of epilepsy. So one of the sort of secondary changes we think is occurring in this model is that we have 3 different animals here. So this is a control animal. This is a PTEN knockout animal, and this is another PTEN knockout animal. Both of these animals have epilepsy. But this animal that has epilepsy has no mossy fiber sprouting. So it's a little hard to make out. But see, there's a blue band of staining here, which is indicative of these axons projecting back into the terminal field. And what that tells us is that in this model, you can get epilepsy without this particular pathology. It's not required for the seizures. But as these animals progress, they start to develop this mossy fiber sprouting. And one of the things we'd like to understand is, what does this change actually mean? So how does having these abnormal circuitry here affect either the seizures in the animal? So when this happens, do the seizures get worse? So we don't know yet. Do other pathologies get worse? So perhaps this is affecting cognition or some other function of the circuit that could be important. So that's definitely a secondary change in the sense that this appears to take some time to develop and is not directly mediated by these abnormal cells. And another aspect of this change that's very interesting is if you look at the cells that actually participate in the mossy fiber sprouting, we see that it's the PTEN knockout cells, which is not too surprising, but we also see normal granule cells start to contribute to the mossy fiber sprouting as well. So that's shown here. So what you're looking at in red is the GFP-labeled PTEN knockout cells. And so this is a panel of the GFP staining in this intermolecular region where the sprouting occurs. These blue dots is a marker for these presynaptic terminals of the granule cells. And if you merge the 2 images together, you see that these blue dots in many cases line up with the red fibers. So that's mossy fiber sprouting coming from one of those PTEN knockout cells. But if you look at these animals, you also see a lot of blue dots shown by these blue arrows here that don't have GFP expression. So that's an indication that that was a normal granule cell. So it's got PTEN, but it's now contributing to this mossy fiber sprouting. So this is sort of first evidence that having these abnormal granule cells in the brains of these animals is now starting to affect their neighbors so that they're starting to contribute to the pathology. We find that very interesting and something we want to look at further. So the final thing we're going to try to do with these animals is, again, using our genetic techniques to selectively ablate these PTEN knockout cells. So because we sort of have genetic control of these cells, we can do a lot of different things to them. And in this case, we've made them express the simian form of the diphtheria toxin receptor. So it turns out mice are pretty much immune to diphtheria toxin. People aren't, which is why we get sick. But if you make these granule cells express the simian diphtheria toxin receptor, that makes just these cells susceptible. So now we can label these cells with this receptor, allow the animals to get epilepsy, treat the animals with diphtheria toxin, which for the most part doesn't hurt the mouse because they're immune, but these cells will die. And that works, so that's shown here. So this is a PTEN knockout animal treated with saline, and the blue is this actual receptor. And then it's very dark here, you can't see the cells, but all these blue cells are gone. So we're able to kill them off selectively in an animal at sort of any time point we want. And some of the initial experiments we've done just as proof of principle is we treated one of these animals with saline, so it starts to have seizures. You have an example of a seizure here. And we treated another animal very early on, just as a proof of principle with diphtheria toxin, and we're able to rescue that animal so it doesn't develop epilepsy. And we're doing experiments now to see how long we can wait to kill off the cells to rescue them. So it could be that at any stage in this disease, these PTEN knockout cells are causing the epilepsy, and if you get rid of them, the animal will get better. Alternatively, it could be because of these secondary changes that you kill off these abnormal cells, but because the normal cells are now participating, like the mossy fiber sprouting I showed you, that it's too late and the animal still has seizures. And don't have conclusive data yet, but we had a couple animals where we treated with diphtheria toxin, and they continued to have seizures. So that suggests that the seizure focus is now spreading beyond just these PTEN knockout cells, and other parts of the brain that were initially normal now contributing to the epileptic focus. So to kind of summarize the whole story of what we think may be happening here in epilepsy is that we think it starts with some kind of brain injury, could be head trauma, genetic defect, something that's altering the neurogenic niche where these newborn cells are being generated. And that leads to these cells integrating abnormally into the brain. And that might occur for some period of time asymptomatically. And that could correspond to the latent period where you have this injury and then years go by before the patient shows up in the clinic with the first seizure. But you could imagine, and sort of our data would suggest that this could be the case, that if you're accumulating these abnormal cells over time, at some point you reach a threshold level where you can start to have seizures. And then you start having seizures, and this could be progressive because seizures are going to disrupt brain activity, which might further disrupt this neurogenic niche. So you continue to accumulate abnormal granule cells, and that would fit with this idea from the last century that seizures beget seizures. So clinically, there's a clinician, a neurologist named William Gyrus, who observed that as people started to have seizures, some patients would have more seizures, and the idea was that as you start to get seizures, the seizures are actually sort of driving the epileptogenic process itself. And this might be one mechanism where they could do that. I'm going to stop there, just thanking some of the people who did the work. So Ray Pun in my lab is our electrophysiologist and did a lot of the physiology and the PTEN work. Candy did a lot of the imaging work. Mike did the correlation studies. Isaiah's done a lot of the EEG work, looking at seizures in the animals. Bethany's done a lot of the stuff with the ablation, which I think is a very interesting avenue. So I'll take questions, and thank you for your attention.
Is there any role for reduced inhibition in any of these models? Or
absolutely.
Neurons?
So I'm presenting one story sort of how we think what may be happening here, and these adult-generated cells may be are contributing to epileptogenesis. I certainly wouldn't say that they're the only way to get to epilepsy. So we know that a lot of inhibitory cells die in the epileptic brain. That's one of the first things that happens in the status epilepticus models. So that's probably important. There's a lot of inflammatory changes, which may be important. There's changes. Can be gliosis and other changes. So I think probably there's many different types of epilepsy. There's 200 plus different epilepsy syndromes. This particular pathology may be important for temporal lobe epilepsy, and it may contribute. But in any particular patient, you may have sort of a range of pathology that actually accounts for their particular seizure phenotype. So in one patient, you may have a lot of abnormal granule cells. In another patient, you may have more cell death. And one of the things we don't really understand in epilepsy is you get a patient in the clinic, and clinicians have sort of their favorite drug that they might start with, but we don't really have clear guidance for a lot of epilepsy conditions is which drug is going to work best in any particular patient. And what I sort of think could be the case is that depending on the particular pathology that patient has, that may determine whether drug A works better than drug B. So I think this neurogenesis is probably part of the story, but it's not going to be the whole story.
Have you considered what other effects rapamycin might have independent of mTOR, since it's a profound inhibitor of immune cells? Right.
So the mTOR story is interesting. Rapamycin, some of the animal models, it seems to work. So Paul Buckmaster has done some studies in mice and hasn't gotten it to work in the mouse models of pilocarpine epileptogenesis. So we don't really understand why that's the case yet. It's a very good point that so there are other effects of rapamycin. So the inflammation is a big one that people are looking at, and I think it could be the case. I mean, we don't really know yet. There's also some anticonvulsant effects of rapamycin that could be important. So my other R01 that gets scored in a week is trying to figure out the mTOR story a little more to figure out is it really mTOR that's pivotal for blocking epileptogenesis, or is rapamycin working by one of these other mechanisms? I don't think we know yet.
Also, if it's an antibiotic, it would affect the gut bacteria.
Right.
And the microbiota have been reported to be involved in autism in the recent paper, so it's a very interesting.
Yeah. So our approach is to go after the gene directly and manipulate mTOR without using rapamycin. So that sort of gets around that problem. Or if they fund it, hopefully they'll get some more definitive answers. Yeah.
Steve.
Oh.
Sorry.
All right. An overarching question is, and by the way, a very elegant
Thank you.
discussion, a very elegant research you've accomplished. But my overarching question is how closely you think these models can be applied to human epilepsy, and some specific components of that question would be use classic techniques of kainic acid and pilocarpine. You know, is this truly a human epilepsy?
Right.
And another example is the dentate story. Since mossy fiber sprouting has been identified in animal epilepsy and humans, it's been, it's been very implicated in the etiology of the epilepsy. But truly, is it a chicken or an egg phenomenon? And one example is in the human world, when we remove a severely sclerotic hippocampus, the dentate layer is often gone.
Mm-hmm.
And you'd think if the dentate was the focus of the epilepsy, if the neuronal pool of dentate neurons is wiped out, that epilepsy should abate, but it doesn't. That's not how it happens in human epilepsy. So these are just some of the kind of mechanistic problems I have applying this model to humans. I wonder if you could comment.
So on the first question on animal models, so this is a perennial fight in the epilepsy field as to what is the best animal model for mimicking human epilepsy. We're not, as I said, it's an ongoing battle as to which is the best model. Is the pilocarpine model that we use is one of the popular ones because it recapitulates some of the features of human epilepsy. But I mean, whether this will really translate well or not, I think we don't know. I think the way we sort of approach models depends on the specific question you're asking. So in our case, we're looking at neurogenesis. Pilocarpine does a very good job of producing these abnormal cells, which is sort of the readout that we want to look at. So I think it's very useful for trying to understand how epileptogenic insults affect that particular cell population. We do know that neurogenesis is disrupted in human epilepsy, so I think in some regard that's probably relevant. But I think in the end, how translatable it is still an open question. So the second question on the mossy fiber sprouting. So it's, for those of you who don't know, it was sort of initially observed in the 80s, and it's sort of one of the hallmark pathologies of human temporal lobe epilepsy. You almost always see it. But it's highly controversial as to whether it actually has anything to do with the seizures or whether it's entirely an epiphenomenon. And so there's sort of the literature is kind of leaning that it could just be an epiphenomenon. Again, we don't know for sure yet. So in our model, you can certainly get lots of robust seizures without it. So this suggests it's not required for epilepsy. It's hard to imagine that a brain with lots of mossy fiber sprouting functions the same way as one without it. So what actually it's doing, we don't know. I think some of these pathologies, it could be that they don't really contribute to the seizures, but they're contributing to the cognitive deficits or the depression or some other comorbidity that's associated with epilepsy because you have a disrupted circuit now. So on your sort of third point that the dentate is damaged in a lot of patients who are getting hippocampal resection, so that raises the question, is that really important or not? So I think, again, that's an interesting story, and it sort of gets to our progression question. So I don't think of epilepsy as during the course of a patient who has epilepsy, I think the disease probably changes over time. And where the seizures originate and how the seizures manifest are probably not the same in a patient who's just been diagnosed with epilepsy versus somebody who's had intractable epilepsy for a long time and now is up for surgery. So it could be the case that the dentate might be important early on in the disease, and then as the disease progresses, that you get progressive damage to the dentate, and it becomes less important, and now the seizure focus is somewhere else in the brain. So that's kind of the questions we'd like to get at. I don't know the answer yet, and again, it could be that it may be it's important for some epilepsies and not others. That would be entirely believable, and certainly for cortical epilepsies, we don't think the hippocampus is going to be all that important. Greg, you had a question?
Yeah. So first off, just in answer to the translation, at least for your work in activating the PI3 kinase pathway, there's an evolving story in the literature that suggests that at least for subgroups of patients with epilepsy and possibly autism and epilepsy, that this work actually might be.
Yeah, that's absolutely right. I didn't focus on the mTOR story.
The question I had was the secondary cells. You're talking about progression here in response to the last question. What do people think is happening in those secondary cells that are getting excited? What sorts of cellular pathology are people seeing, or electrophysiology-wise?
So we don't really know yet. We've started to look at that. So with this model, we're recording from PTEN knockout cells, but that's tissue we can also record from some of the neighboring cells that are PTEN, they have normal PTEN expression. And so far, we haven't picked up big differences. So it's easy to tell the knockout cells from the neighboring wild-type cells. So far, the wild-type cells seem to behave pretty much like wild-type cells in a control animal that doesn't have epilepsy. That's one of the things we're very interested in finding out. So you could certainly imagine as sprouting occurs, that these neighboring cells maybe start to receive more input or show other changes indicative of sort of the epileptogenesis in an animal. But I think at this point we don't have a good handle on it. So one of the things we explored with the mossy fiber sprouting is one of the ideas is that you get mossy cell death. So there's a population of interneuron called a mossy cell in hilus, and they project to the region where the mossy fiber sprouting occurs. And in a lot of these epilepsy models, in humans with epilepsy, these mossy cells die, which sort of de-innervates this region. And that's thought to possibly be the stimulus for why the granule cells then sprout to sort of take up this empty space. So we looked at our PTEN models to see if we have robust mossy cell death. And so far, we're not seeing it. It looks like the mossy cells are still there, which suggests that there's some other stimulus that's provoking this mossy fiber sprouting from the PTEN knockout cells and the wild-type cells next door to them.
We have to wrap it up. Thank you very much.
Thank you.
For an exciting talk.